Okay, last unit or last chapter on our immune system, chapter 15. This is looking at the specific immune response and immunization. So this will complete all of the immunization, chapter 13, chapter 14, and chapter 15 is what is collected in this unit. So let's go ahead and begin with our specific acquired immunity. Your specific immunity is basically your third line of defense. This is the line of defense that is going to be a specific response to a specific targeted organism. It's going to be acquired after you've had some sort of immunizing dose, either through infection or through vaccination. And this is what's going to give your body the advantage against the microorganism, because you are going to get sick the first time, but after that, your third line of defense or your specific immunity is going to remember this microbe on a subsequent occasion. And so you are going to get sick once with this, or a very short amount of times, but your body is going to remember it, and it will fight it much more quickly when you see it a second time. We're going to talk about something called immunocompetence. Immunocompetence is essentially the ability for the body to react to some sort of substance or organism that it deems as foreign. When it sees these foreign substances, you're going to begin with the development of our B cells and our T cells. Remember, back in our second line of defense, our non-specific defense, we had those granulocytes, those basophils, eosinophils, and neutrophils, and those all got triggered when we talked about phagocytosis and inflammation and those kinds of things. But we didn't ever really talk about the other side of that graph, of that picture of the blood cells. We've got our B cells and our T cells. Now in our third line of defense, when we have the initiation of our immune system, we see those foreign substances, we're going to go with the development of our B cells and our T cells. The lymphocytes are going to become specialized. They're going to react to a specific antigen, or what we call a specific immunogen, and it's going to target that specific substance and nothing else. There are two characterizations in the third line of defense that we don't see in any of the other two lines of defense. The first one is specificity, that target to that one specific antigen. And we also see something called memory. Again, I talked a little earlier about that on a subsequent exposure, the body remembers that antigen and is going to target it, and it's going to target it very rapidly and very efficiently, oftentimes when we don't even know we've been sick. So you can have a very rapid memory response the second time around. There are 5 stages to development and interaction of this third line of defense. The first thing we're going to have to do once we've been exposed to a particular antigen is the development and differentiation of those B cells and those T cells. Once we see the development of those B cells and T cells, we're going to have, or what's going to initiate the development of those B cells and T cells is the presentation of an antigen. We're going to talk specifically here in a little bit about antigen presenting cells, or antigen presentation, and this is what is going to initiate the activation or the challenge of our B lymphocytes and our T lymphocytes. Then once we've challenged those B lymphocytes and T lymphocytes, the B lymphocytes are going to begin to produce something called antibodies, and the T lymphocytes are going to have their own specific cellular response to those organisms. Let's do a quick overview of the whole lymphatic system. We've got the B cells and the T cells, and these are going to work together to initiate that specific response. There's a significant amount of communication between the two types of cells. As a matter of fact, we have to activate a specific B cell, or T cell, before we can activate any of our B cells. Once we have antigen presentation, both the B cells and the T cells are going to be activated, and they're going to target the organism or the antigen that is being presented to them by the immune system. Here is a quick step-by-step graphic of the whole immune response, the steps in your acquired immunity. The first thing is you're going to have lymphocyte development. We're going to talk about something called clonal selection and clonal expansion, and that is going to be our lymphocyte development. Then you're going to have presentation of the antigen. Once that antigen is presented to the B cells and the T cells, they will be challenged, and which means that they're going to become mature, they're going to become activated so that they can be functional B cells and T cells. Then once we have the challenge of the B cells, those B cells are going to produce antibodies, and then the challenge to the T cells will initiate something called your cell-mediated immunity. When we have an immune response, we have those organisms getting into the body, into the cells, into the tissues, and being presented as foreign to the immune system. Some cells are going to have to cross the first line of defense, and they're going to enter into the tissues. They're going to have phagocytes migrate to the area of entrance where they are going to consume that antigen. Those phagocytic cells that consume those antigens are what we call antigen-presenting cells. You have two types of antigen-presenting cells. You've got your macrophages and your dendritic cells. The macrophages are going to ingest the pathogen, they're going to consume it, and they're going to present a portion of that on the surface of the macrophage. And then the rest of the immune system will recognize that and be triggered to start. And we'll get into the specifics of that antigen presentation here in a little bit. The second antigen presenting cell is going to be the dendritic cell. These are going to also ingest antigens. They're going to migrate to the nearest lymphoid organ, the spleen, a lymph node, something like that. And there they're going to begin that antigen presentation in that lymphoid tissue. Once you have antigen presentation, it's going to present it to a specific T lymphocyte called the T helper cell. And that T helper cell then is going to activate the B cells. That T lymphocyte is going to recognize the piece of that antigen that has been exposed on that antigen presenting cell, and it's going to activate the B cells. If we look at this picture in the textbook, this picture I believe is on page 400 and, let me double check here, 427. And if you will have this book open, or this page open in your book while we're going through a good portion of this, this antigen presentation and activation, I think it will help you understand what's going on. Once you have your B cells and your T cells, and they're going to mature, your B cells are going to mature in one place, your T cells are going to mature in another, and we'll talk about where those mature here in a little bit. But let's move down to the second section here, where it says antigen contact. We're going to have antigen presentation here. You've got your dendritic cell that is going to consume the antigen. It's going to consume it and present it on a cell surface marker that the cell will recognize. And now you've got your T cell here that is going to recognize that this is a foreign substance, and it's going to bind to that dendritic cell, and that T cell now is going to send a signal to our B cells, and those B cells then will be activated to differentiate, and we're going to then, as an end product, make antibodies. This is what we call our humoral immunity. The T cell, or excuse me, the dendritic cell that has or the macrophage, depending on your antigen presenting cell. This one just happens to be a dendritic cell. I've lost my little cursor here. There you go. You've still got this antigen that's presented onto the surface. It's going to activate that T cell, and that T cell then is also going to activate other T cells to divide and differentiate and become what we call memory cells or effector cells. Okay, and these effector cells are going to have an effect on the antigen and will destroy it. So depending on the cell, whether it's a T cell or a B cell, you're going to have activation of those T cells and those B cells when you have antigen presentation. And we're going to talk about each one of these specifically step by step. So follow the picture and then as we move on, we will go a little bit further into the discussion of antigen presentation and activation. When you have B cells and T cells that are going to be challenged, or excuse me, when you have the immune system being challenged by an antigen, and the B cells and the T cells are challenged by that antigen, those B cells and T cells are going to proliferate and differentiate. What I mean by proliferation is that they are going to create clones of themselves. And when they create clones of themselves, these cells are genetically identical. Every single one of them has some sort of, they are genetically the same from cell to cell to cell. And they are going to remember the same antigen over and over and over again. These are going to differentiate in that some of them will become effector cells, and some of them will become memory cells. B cells have effector cells that are called plasma cells, and then B memory cells. And then T cells have effector cells that are like cytotoxic T cells, T helper cells, and some become memory cells. So they're going to proliferate and they're going to differentiate. So this is what we call clonal expansion. Let's talk about the B cells specifically. Let's take a really quick overview here of what the B cells are and antibodies that get provided or produced by these B cells. When we have the activation of our B cells, the offspring of those B cells are called plasma cells. Some of those clones are called plasma cells. These are the effector cells of our humoral or B cell mediated immunity. These are going to be programmed to produce and secrete something called an antibody. An antibody is a protein that will be found in tissue fluids or in the serum fraction of the blood. When these antibodies find the antigen that they are programmed to, they're going to bind to that antigen and they're going to mark that antigen for destruction or for neutralization by some mechanism like complement fixation. This B cell activation is part of our humoral immunity. We can divide our specific immunity into our humoral, which is B cells, and our cellular, which are T cells. So our B cell immunity is our humoral immunity. T cells, on the other hand, when they are activated by an antigen, they're going to give you 3 different types of progeny. Outside of the memory cell, you're going to have 3 different types of effector cells. You're going to have T helper 1 cells, and these are going to activate macrophages to help with the activation of another type of cell called a cytotoxic T cell. Then you're going to have T helper 2 cells. These are going to assist in the B cell process. These are the cells that are going to wake up those B cells to activate and produce antibody. And then you have your cytotoxic T cells. These are going to lead to the destruction of these antigens by binding to them and producing toxic enzymes that are essentially going to poke holes and shred apart the antigen by itself. So there's no antibody here. This cell is going to target the antigen specifically and deal with it on its own. So let's look at this. That was kind of real quick, down and dirty about the whole activation of your third line of defense. Let's go back to the very beginning and start talking about that lymphocyte development and how they begin to develop and where they go once they develop. If you remember back in chapter 14, where we started talking about your second line of defense, and I showed you that picture of the cells and how they all start with the same stem cell. You've got your hematopoietic stem cell, and the graph that I'm talking about is on page 407 in your textbook. You start at that hematopoietic stem cell. All lymphocytes are going to arise from the same basic stem cell type, and that is that hematopoietic stem cell. You've got part of that, remember, that goes to that pluripotent stem cell and gives us all of our granulocytes, our basophils, our eosinophils, and our neutrophils. But if you go and look to the right side of that picture, you have your lymphocyte cell line, or your agranulocytes. And the macrophages are part of those agranulocytes, But the other two cells that I want to talk about are the lymphocytes, the B cells and the T cells. Those B cells and T cells are going to come from a lymphoid stem cell, and that lymphoid stem cell will allow these cells to divide into our B cells and our T cells. B cells will mature in the bone marrow. They are going to go into the area in the red marrow of the bone, and that's where they're going to mature. And they're then going to move on to the lymphoid tissues like the spleen and the lymph nodes. They're going to migrate to the cortex of the lymph node, and we're going to see that the T cells are also going to go to the lymph nodes in the spleen, but they're going to migrate to the medulla of the lymphoid tissues. The T cells do not, they may start in the bone marrow, but they travel to the thymus. That thymus is that big organ that sits right on the surface of the heart, and they're going to mature in the thymus and then find their way to the lymph nodes and to the spleen, to these lymphoid organs. And here they're going to find their way into the medulla of these structures. One of the characteristics about the immune system is that whether it's your first, second, or third lines of defense, you have to understand, or the body has to understand, to be able to recognize self versus non-self tissue. These B cells and these T cells, when they go and target a cell, they have to know whether they're targeting a self cell or a non-self cell. And one of the major ways that they do that is through cell surface markers. One of the main functions of the immune system and these markers is to help with the attachment of these B cells and T cells to non-self or foreign antigens, and to leave self markers alone. These self markers are also going to bind to receptors that are going to indicate self versus non-self, and we're going to talk about some specific self surface markers, which are called MHC, or major histocompatibility complex markers. These markers on the cell surface can also receive and transmit chemical signals that are going to coordinate a response between our B cells and our T cells, and they're also going to help in the cellular development, aiding in the production or the development of the B cells and the T cells. Let's talk about the MHC cell surface receptors. This is called the major histocompatibility complex system. If you think about what the word histo means, if we're talking about histology, that's the study of tissues. So tissue compatibility, pardon me. These are a set of genes that are coded for on the human cell that are going to give us these cell surface receptors. And these are going to be these proteins, they're glycoproteins, that are going to be found on every cell that has a nucleus. So therefore they're not going to be on the red blood cell, but every other cell in the body has these cell surface markers. This is also known as the human leukocyte antigen system, or the HLA system, but you're going to find your book talks about them as the MHC markers. There are three classes of MHC genes that code for specific cell surface markers. You've got class 1, class 2, and class 3. These are going to be markers that are going to basically determine self from non-self tissue. Class 1 genes are going to code for markers that display characteristics of self on every cell. Every cell that has a nucleus will have a class 1 marker on it, and this will tell tissue from self from non-self tissue. It's not necessarily going to be blood cells only. So these are going to be found on tissue cells of any kind that have a nucleus. Class 2 genes code for receptors that are going to be found on macrophages, dendritic cells, and B cells, and these are going to be involved in the presentation of antigens to our T cells when we initiate our B cells and our T cells to begin differentiation and development. So these class 2 are only going to be found on these three different types of cell, these antigen presenting cells and the B cells. Class 3 genes are going to encode for proteins that are involved in the complement system. Remember back in chapter 14 we talked about complement fixation, and that these cells are going to have the genes that code for the antibodies that bind to those cell surface, those class 3 genes, and they're going to initiate the complement system. If we look here, we'll see the class 2 and class 1 and class 2 markers. We're not going to really talk about class 3 markers much. We're going to spend our time on the class 1 and class 2 markers. These are going to be found on host cells, and if they are not part of the host cell makeup, these are going to be tagged for destruction. Because if they're absent, then the immune system says, I don't recognize these cells as self-tissue. This is non-self tissue. It's foreign. I'm going to destroy it. These are going to be used to present antigens, particularly these class 2 markers. If you look here, you'll see that these class 1 markers, they're found on any nucleated human cell. You've got basically a protein that goes through the membrane, attaches it to the membrane. You have an antigen binding site here where that antigen is going to bind, and this is going to initiate destruction of a cell that is going to, or the antigen that is going to be caught in this antigen binding site. If we look at the class 2 MHC protein, you'll see you also have an antigen binding site. It's also bound into the membrane, but this is going to be found on the dendritic cells, the macrophages, and some of our B cells. This is going to be on all the cells. The class 2 are going to be only on the 3, macrophage, dendritic, and B cells. On those antigen presenting cells and the cells with those MHC1 proteins, we can bind our antigen to those, but until we can bind our B cells and our T cells to those, there's not going to be any destruction. So when I have my antigen presenting cell on my B cells and my T cells, they have to have receptors that will also bind to those antigens. So the B cells are going to have receptors that bind to the antigens, and the T cells are going to have receptors that are going to bind to those complexes as well. All of these receptors are going to bind to specific antigens, and you're going to have a specific response. And because antigens are very diverse, there are tons of different antigens or different possibilities. You're going to have a number of unique lymphocyte receptors that have to exist at any given time, and this is what we call clonal selection. Think about this. A baby, before it's born, is in this very sterile, very protected area. Have you ever wondered why when a baby takes its first breath, it doesn't get sick? It doesn't get sick because it has already within it, coded on its chromosomes, these MHC, major histocompatibility complex receptors. So when antigens come in, they bind to those, and our B cells and our T cells also have receptors, and those B cell receptors and those T cell receptors are going to be able to bind to those MHC antigen complexes, and we're going to have an immune response right away. Our immune system is going to start working right away, and we've got this thing called clonal selection, which allows us to select the appropriate receptor for the appropriate antigen, and we will have basically a genetic immunocompetency right from the very get-go. So we have our B cells and our T cells, we have our MHC receptors, and all of these things have to work in harmony. But they can't do anything until we have some maturation of our major fighters in this immune system, which are our B cells and our T cells. Let's go back. We said that the bone marrow, or excuse me, the B cells they would mature in the bone marrow, and they're going to, think about the bone marrow as a nursery. The bone marrow is a site that has a number of cells called stromal cells. These are very large cells, and they basically are there to nurture the maturation of B cell development. They're going to provide chemical signals that are going to turn on that B cell development when we need it. We certainly don't want more B cells than we need, because that can be dangerous. That can be a cancer, B cell lymphomas. And so we have to turn these on only when we need them and keep them turned off when we don't. And these stromal cells are going to act kind of like a nanny in this B cell nursery that's only going to allow those that are properly ready to be initiated into development. Once those B cells have been initiated, they're going to circulate through the blood, and they're going to move to specific lymphoid tissue, to specific places in particularly the lymph nodes, the cortex of the lymph nodes. They're going to move to the spleen, and they're going to move to that gut-associated lymphoid tissue. Once there, they're going to bind to molecules that they come in contact with the appropriate antigens throughout the life of the person. So you've got these B cells fixed in tissues that are ready for whatever antigen they were programmed for, and they will come into contact with those antigens throughout the lifetime of the human. Once B cells are in their particular tissues, they will be able to identify their particular markers by the fact that they have antibodies as their cell surface receptors. So those antibodies, particularly IgM and IgD antibodies, are going to help with antigen recognition. The T cells are going to mature a little bit differently than the B cells. The B cells have their cell surface receptors are antibodies. The T cells will mature in the thymus, and when they begin to mature at the expression of hormones within the thymus, they're going to produce what are called CD4 or CD8 co-receptors. If it has a CD4 receptor, it will bind to MHC class II major histocompatibility complexes, and it will turn on that T helper cell. If it has a CD8 receptor, it binds to MHC class I, which are going to be found on what are called the cytotoxic T cells. And you're going to have these cells constantly circulating through the lymphatics, through the generalized circulation, and they're going to migrate to specific areas also in the lymphoid tissue, particularly the medulla of the lymph nodes, and then into the spleen. So if you go back to the picture on page 427, you're going to see that your B cells and your T cells have specific receptors on their surface. If you look at that picture, you'll see these little Y-shaped structures on the surface of the B cell, and those are the B cell or those are the antibodies. Then you have on the T cells, you just have these kind of straight lines. Those are your CD4 and your CD8 receptors. These receptors, whether they're CD4, CD8 receptors, or your antibodies, are going to bind to the antigen presenting cell once they have bound to the MHC antigen complex. This is a good table. I just thought it was one worth putting in here so you could see the difference between B cells and T cells, where you find them as far as the structures of the lymphatic organs. Do they circulate in the blood? What are their general functions and the like? Take some time to look at this to try and help you understand what's going on. And we can, if you have any questions, please email me so that I can be sure to answer them and make sure you understand the difference between B cells and T cells. It sounds like I'm doing a lot of repeating of myself with this cellular development. What I want you to understand, particularly with this picture, you'll see that both of these cells are going to come from the bone marrow. B cells are going to remain with the bone marrow. They're going to migrate to the red marrow. They're going to be directed by those stromal cells. They're going to initiate maturation, where they will receive their cell surface receptors, which are these antibodies. If they're T cells, those T cells are going to migrate to the thymus. Once they're in the thymus, they'll receive their CD4 or CD8 receptors, and in both cases, they're going to move to the lymphoid organs. Here in the lymph node, the B cells are going to be in the cortex, the T cells are going to be in the follicles within the medulla, And all of these are what we call immature or naive cells. These are going to be cells that are just waiting for their antigen, for their the particular microbe that they are designed for to come and turn them on so that they can begin the whole process of killing. When you have all of these cells, now that we've given them their receptors and stuff, we need to move on to this thing called clonal selection and clonal expansion. This is essentially the second stage of development. We've moved these cells. You've got to remember, we've done all of this stuff, but all of these cells are still immature, and we need to be able to stimulate them to activate. And this is where this second stage of clonal selection and clonal expansion comes. You're going to have the stimulation by an antigen. And when that antigen comes in contact with those B cells or those T cells, when it comes in contact with those clones, it's going to go through, those B cells and T cells are going to go through mitosis. And when they do that, they are going to begin to produce a huge number of cells. If you will go to the figure on page 431, you will see that there is a picture of this lymphoid stem cell that is beginning to go through clonal selection and clonal expansion. And we'll continue to move on with this discussion, but if you'll use that picture, it will help you understand what I'm talking about. Here we have, this is the picture on page 431. You have your lymphoid stem cell, whether it's a B cell or a T cell, and it has its particular receptor. It's going to produce this repertoire of clones, okay? Each one of them is going to have a specific receptor, and these receptors are coded for genetically. This is just like those MHC class proteins. These receptors are also going to be genetically distinct. You're going to see here that we have potentially some self cells that are some self receptors, and when we have those self receptors, those will be eliminated. Those clones will be eliminated, so we don't have this repertoire that includes cells that are going to target our own self tissues. All of these now have unique receptors that are going to be directed towards a particular antigen. Once that antigen is displayed, once we have the entry of that antigen, we're going to select the particular clone, okay? Once we select this particular clone, that the antigen, this antigen binds only to this clone, we are going to have this massive clonal expansion where we're going to have a number of these that are going to begin to divide and differentiate, and we're going to have an immune response now against this antigen. This is what I was talking about when I was talking a little earlier about this whole number of cells that we have that are ready for antigen entry and the immune response against that particular antigen right away. All of these are genetically defined through our through the DNA within our cells. So we have this immune response that's ready to go as soon as we have some sort of challenge to that immune response. We can divide clonal selection between the antigen independent period, which is essentially this period where we are going in and making clones of these lymphoid stem cells and eliminating any sort of self cell. Once we have these lymphocyte clones, now we can go into the antigen dependent period, where only the clone that is selected will begin to divide and differentiate. The rest of them are going to sit there, and if they never get challenged, they will never get triggered. And so this proliferation then will result by this antigen triggering the single clone, and all of its clones that comes from mitosis are going to have the exact same antigen specific receptor and will bind only to that antigen. When you saw that picture before, one of the things that it did was we showed that any clone that has a self receptor is going to be destroyed during that that beginning antigen independent period of clonal selection. This is going to be what they call clonal deletion, where you can have those self receptors, those cells are going to be removed and you're not going to be attacking your own tissues. The autoimmune diseases that we know of are thought to be caused by a loss of the tolerance of self versus non-self. We've got those cells that don't get deleted through that whole clonal process, and we end up with cells that have self receptors that are going to go and then start damaging self tissues. Clonal deletion is, again, going to be that self-recognizing clones, and they're going to be eliminating those. So all of those will get destroyed in this process, and we don't have to wait for any type of antigen dependent period. It's going to destroy them right from the very beginning, so that the self tissues are not going to be harmed. So let's talk about antibodies. These antibodies, I said that our B cell receptors have antibodies the B cells have antibodies on their surface that act as cell surface receptors. These receptors are going to be what we call immunoglobulins, and these are large proteins that are going to be antibodies that are going to be secreted by B cells. They're also going to act as antigen receptors of the B cells. These are a very large protein, about 1,200 amino acids in size, and they have this very peculiar Y shape. You've got 2 heavy chains and 2 light chains, and those heavy chains and those light chains are going to make this peculiar Y section. At the top of each Y, at the fork of the Y, they have this pocket that's called an antigen binding site. And that antigen binding site is going to be variable to the shape of the antigen that this antibody is created for. And that antibody then is only going to bind to a particular antigen. When we look at the antibodies, you're going to see that they are divided into what are called the variable regions and the constant regions. And we'll look and see what each of those means here. If you look here, you've got your, this portion of the picture. I don't really care if you recognize that you have 4 different sections along a chromosome that the heavy chain comes from and 3 different that the light chain comes from. What I want you to pay attention to is this picture right here. You've got your 2 heavy chains and your 2 light chains. Each heavy and each light chain have a variable region, and each heavy and each light chain have a constant region. This variable region is going to be the antigen binding area, and it's going to be unique to every antigen. This constant region is going to be the same for every antibody of a particular class. We have 5 classes of antibodies, IgA, IgM, IgG, IgE, and IgD. And let's say we have 100 IgG protein or antibodies. This constant region is going to be the same for every one of them. Their variable region will be different, but the constant region is the same for every IgG antibody. These heavy and light chains are going to be held together by disulfide bridges, and it's this portion that binds to the antigen, and this constant region here, if it's acting as a receptor on a B cell, it will bind to that receptor with this on that B cell with this constant region. T cells also have receptors, so CD4 receptors are going to bind to the MHC proteins, but the antigen receptors, much like the antibodies, are going to have antigen binding sites. They're also going to have variable regions, and they're also going to have constant regions. Unlike antibodies that are secreted into the bloodstream, the T cell receptors are never secreted. They are bound to the cell surface and will never be secreted outside of the cell. It will always be attached to the cell and will remain there. So we have our B cells and our T cells have been produced, they've been initiated, and they've been through the process of maturation. Now what we need to do is we need to present the antigen. to initiate the immune response, you have to have an antigen that provokes the immune response. Any antigen is a substance that's going to cause an immune reaction. It's going to be perceived as foreign by the immune system, and it can be any molecule that is organic and degradable. If you look here, we have proteins, lipoproteins, glycoproteins, nucleoproteins, polysaccharides, lipopolysaccharides, polypeptides. If it can be degraded, it can act as an antigen. One of the characteristics of an antigen is that they are very complex, and when they are complex, they can grab the attention of the immune system, not like these smaller molecules, that we would, small, say you have a sodium ion or a sodium chloride ion or a small sugar molecule, or if you have these very long repetitive molecules, these are going to have a much less antigenic effect. These are going to be things that the body doesn't necessarily recognize as foreign because they aren't attached to complexities like cell walls, cell membranes, things like that. So antigens tend to be very complex, and they are going to be more immunogenic than when we have some smaller things like small polymers or these smaller molecules or atoms. When you have something that is large enough to grab the attention of the immune response, you're going to have cells that are going to act as surveillance cells. These surveillance cells, these are going to be your macrophages and your phagocytic cells. These are going to recognize and respond to a particular portion of the antigen called an epitope. And that epitope is going to be the thing that the immune system recognizes. It's not going to recognize the entire antigen. It's only going to recognize a portion of it. So when you see the immune system reacting, it's only reacting to the epitope, not the antigen completely. Some microbes also have what we call mosaic antigens, where they have a number of different epitopes on their surfaces, and you can have a number of different lymphocyte responses, a number of different antibodies binding, a number of different lymphocyte responses. You also have something called a hapten. A hapten is basically the determinant group that causes the reaction. It's basically an epitope without the rest of the molecule. It's too small to be immunogenic, so it doesn't elicit an immune response. But if we link it to a larger molecule, if we put it with a carrier molecule, it can then cause some development of an immunological response. If you look at this picture here, this picture is on page 433 in your book. This is essentially a hapten. It's a very small molecule, and if we were to inject this into this mouse would not produce any antibody response. But if we bound this hapten to a larger molecule where it was now visible to be seen by the immune response, when we inject that into this mouse now is going to form an antibody to this particular hapten. Notice that it doesn't produce the antibody to the carrier molecule, but to the hapten. So this is the way you can actually make antibodies in the laboratory. If you think something is particularly or could possibly be antigenic, bind it to some inert molecule, you put it into a mouse, and you produce what is called a monoclonal antibody. You can also put them in rabbits. They're not quite as defined when you put them in rabbits. They're called polyclonal antibodies at that point. But anyway, you have the antibody produced. This is an animation. I'm going to see if I can get it to play. I was having some trouble with it, but I'll see if I can get it to play. If I can't, then I will download these so that you can get to them and watch them on your own.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
So you can see how the epitope is what the antibody recognizes, and you can have that immune response. In a basic immune response, you're going to have that encounter between your B cell or T cell and the antigen. And the antigen is generally going to go through some sort of processing after that encounter. Once you have that antigen binding to that antibody, then you're going to have, or binding to that T cell, that antigen is going to be destroyed or neutralized through some sort of processing. Once we have those antigen presenting cells, they're going to act on those antigens, and they are going to then give some sort of a chemical response that's going to allow for those antibody or antigens to be presented on the surface of those macrophages or dendritics. So remember, our antigen presenting cells are our macrophages. We also have those receptors on our B cells, and then our dendritic cells. Our macrophages are going to have those MHC class 2 receptors. And our dendritic cells are going to have those MHC class 2 receptors, and these are going to be the ones that are going to initiate an immune response. Once they engulf an antigen, they are going to modify it so that they can present it on their surface so that it can be recognized by the B cells and the T cells. Once it's processed, then that antigen will be bound to that MHC protein. If you'll go back to that picture on page 227, you'll see that we have that picture of that dendritic cell. The antigen is being consumed, and you'll see it's got those red MHC markers on its surface. Once that dendritic cell consumes that antigen, it's going to modify it, and that piece of the antigen, not the entire antigen, just the determinant portion, is going to be bound to the MHC receptor. Once it moves to the surface of that MHC receptor, or onto the surface of that antigen presenting cell, it is now accessible to that T helper cell, the T lymphocyte, that's going to turn on both our B cells and our T cells. The antigen presenting cell will activate that CD4 T helper cell that's in the lymph node, and these are going to bind to these T cell receptors, and they're going to bind to the MHC protein. They're going to bind to the piece of antigen that it's bound to, and then the T cell receptor is going to bind to that whole complex. If you look at the picture on page 427, if you move over to the dendritic cell that is to the left hand side, where that T cell has bound to the MHC antigen complex, you will see you've got that T helper cell, and it's got an arrow pointing over to the B cells. Once that T helper cell binds to the antigen presenting cell, it's going to then activate that T helper cell, and that T helper cell is going to produce a chemical called interleukin 2. That interleukin 2 is going to be like an alarm clock to the B cells. When that alarm clock goes off, it's going to now activate those B cells, and those B cells then are going to divide and differentiate into their memory cells and into their effector cells. So if you look here, we have our antigen, our foreign microbe. It gets consumed through phagocytosis, and a portion of that antibody, or excuse me, that antigen is going to be bound to this MHC class 2 receptor. If we come and look at this larger cutout here, here are the pieces of the antigen, and they're going to migrate to this MHC protein. Once they're bound to that MHC protein now is going to exist now on the surface of this dendritic cell. Once that happens, now we have this T cell here. That T cell is a T helper cell that binds to this MHC class 2. this antigen, and then the CD4 receptor. Let's look here. Here's a large cutout. We've got our antigen presenting cell with our MHC class II, the antigen, and then our CD4 receptor that binds to the whole process here. This now is going to connect the antigen presenting cell with the T helper cell. This T helper cell is going to have some jobs now to do. It becomes activated. It produces those interleukins, and now when it releases that interleukin 2, it's going to link it to the B cells, and those B cells are going to be turned on, and we're going to start that whole process of cellular differentiation on the B cell side. Now this is just the B cell side. This is just the humoral immune response. Once we have that T helper cell, that T helper cell turns on those B cells by the release of interleukin, and we're going to have clonal selection of the particular receptor cell. It's going to bind to the antigen, and we're going to have that antigen being processed and presented, and that B cell is going to be activated. Once that B cell is activated, it's going to start to proliferate and differentiate, and we're going to have that clonal expansion where we're going to make B cell after B cell after B cell that is specific to that particular antigen. Those B cells are then going to begin to produce antibody, and those antibodies are going to be produced through some cells called plasma cells. I would probably flip-flop these right here. When you have clonal expansion, you're going to have proliferation where we're going to have lots of these cells over and over and over again, and then some of them are going to differentiate into our plasma cells and our memory cells. The plasma cells then will release antibody, and antibody then will get into the circulation and move those antibodies all throughout the body. So here is our B cell activation. Let's go ahead and start right here. Okay, we've got phagocytosis here. We've got the microbe. It's being presented with this MHC class II marker. So we've got our antigen, and now we've got our T helper cell. That T helper cell is going to be that T helper cell that produces that interleukin, and when it does, it activates this B cell. This B cell now has two pathways that it can go. It can become an effector cell, which is called a plasma cell. This plasma cell is going to release antibody into the bloodstream. If it doesn't become a plasma cell, then it's going to become a memory cell. That memory cell will go through mitosis. We'll have a number of different memory cells that will continue to float through our circulation and remain in the lymphatic circulation for a number of years. So we continue to have this memory over and over and over and over and over again. We said that one of the jobs of our B cells, let me jump back to this picture real quick. Here we have our plasma cells. Those plasma cells are going to release antibodies, and these antibodies are the thing that are going to target the antigen, and they're going to target that antigen to be neutralized or destroyed. And so it's not really the B cells that are doing the work. They are basically the factory, and the factory is going to produce the antibody, and the antibody are the proteins that are going to do the work. So let's talk about antibodies specifically. There are 5 major classes of antibodies. These are very large polypeptide chains. Remember I said you had a heavy chain and a light chain. You have 2 heavy chains and 2 light chains. So you have 2 arms of the antibody. They are connected by covalent bonds called disulfide bonds or disulfide bridges, and they have this antigen binding region, or the Fab region, and then they have this other constant region called the crystallized fragment. This is where you're going to have the binding site to the cell. The variable region is going to be the binding site to the antigen. That Fab site is where the epitope, it recognizes the epitope of the particular antigen. it is going to be what we call hypervariable, meaning that the better the fit, the stronger the antibody response, the stronger the immune response. Now, you can have other antigens binding in there that don't have an exact or perfect fit, and they will give you a mild response. But if you have an exact fit or a perfect fit, you're going to have a very strong response. So you do have some hypervariability there. The crystallized or constant region is going to have that portion that's going to bind to the membrane of the cell, and it's going to be the thing that helps to recognize a particular antigen, and it has an effect on the binding of that cell and the role that that cell is going to play in the immune response. So here is a picture in your book. This picture shows you exactly what is taking place. This picture is on page 437 in your textbook. You have this region here, this antibody binding region, and you've got your antigen and you have your antibody here. Each of these has that specific variable shape. If you have a perfect fit, this is going to initiate a very strong reaction. If you have a poor fit, you're going to have a reaction, but it's going to be relatively weak. But if you have no fit at all, there will be no activation to this B cell response, and you won't have an immune reaction to this particular microbe by this particular antibody. But you will probably have a reaction by some other antibody in the system. You have a number of things that the antibody can do. The antibody can tag bacterial cells. When you tag these bacterial cells, you can increase the effectiveness of things like phagocytosis. When you basically wrap these antibodies around a particular cell, this is called opsonization, and opsonization will allow for these macrophages to engulf these bacteria more readily. This is kind of like giving these macrophages a handle to grab onto. Antibodies can also neutralize viruses. Remember, viruses have those spikes, or they have those adhesion proteins on their surface, and those antibodies will bind to those adhesion proteins, and they will neutralize the virus. Here you would have an enveloped virus, and all of the antibodies will bind to the spikes of the envelope and neutralize that virus. So now they can't bind to tissues, and this is going to tag these, and they're going to be marked for destruction. These can also, antibodies can also bind to what we call the toxicity sites in a number of different toxins, and when they bind up those toxicity sites, they can't bind to the receptor tissues, and the toxin gets destroyed. Here is something called agglutination, and this is where you have the cross-linking between antibodies and a number of different bacteria. So all of these antibodies bind up, and you have this kind of clumping reaction where everything, it just kind of gets all wadded up together, kind of gets chewed together. Every time I think of this, I think about when you put chewing gum in your mouth right after you've eaten, and you kind of have gum and food all mixed together. Here you have bacteria and antibodies all mixed together, and this is going to increase the ability of macrophages and dendritic cells to phagocytize these bacteria. Remember that antibodies also will fix complement. This is basically, they will initiate this complement cascade that will poke those holes in the membrane and lyse the cell. Also, you can use antibodies to precipitate to small, tiny antigens, and these will then, what we call, aggregate, and you can use these for testing. And this is what your pregnancy tests and your rapid strep tests and things like that are all about. Let's look at the particular antibodies as particular classes. You'll see that we've got 5 different classes: G, A, M, D, and E. All of these, except for A and M, are going to act as a single monomer unit. You'll see that the IgA is a dimer and the IgM is a pentamer. This chart gives you some definite characteristics about these, and how much of these are in the blood, what their job is, and the like. But let's go ahead and talk about these one at a time. IgG is the main antibody in circulation. It's about 80% of the antibodies in circulation. This is going to be found in the serum fraction of the blood. It is the only antibody that can cross the placenta, and it does begin the activation of complement. You have two of your antibodies will what we call fix complement or activate that complement cascade that we talked about in chapter 14. This is also the antibody that's going to be initiated on what we call a memory response. I will show you a graph here when we talk about memory versus the initial response. So IgG is the main antibody in circulation. IgA has two different shapes. It has a monomer in serum, but a dimer in secretions. It is secreted by antibody, or it is going to be one of the antibodies that is secreted into mucus, into urine, into tears, breast milk, colostrum. It's about 13% of the antibodies in circulation. It does not fix complement. It does not initiate complement, but it is the most important defense in our mucosal defense mechanism. Any place that you have a mucous membrane, you have an enormous amount of IgA antibody. You're going to find this specifically in breast milk. This is what's going to help with the immune response of that will help take care of the baby after it's born and the mother breastfeeds, because it will gain benefit from these IgA antibodies that are passed from the mother to the child. IgM, this is a pentamer. This is a huge antibody. It has 10 antibody binding sites now instead of just 2. It also circulates in the immune response. It's only about 6% of the antibody in circulation, but because it has 10 binding sites, it can pick up a lot very rapidly. This is going to be the first antibody made in an immune response, whether it's a first response or a second response. It's going to be one of the first ones made during an infection. Because it is so big, it can take up a lot of antigen very rapidly. It can also fix complement. It can turn on that complement cascade. And this is also a very good antibody for agglutination. This can bind up a lot of antigens, and you can pull up a lot of them, and they can be dealt with very quickly. IgD is not found in serum. This is not one that you're going to be able to pull out of the blood when you do an antibody titer. This is going to be found on the surface of the B cells. These are the antibodies that act as those cell surface receptors. There's only about 1% of the antibodies total to be accounted for are the IgD antibodies. And then finally, the IgE antibodies. These are going to be antibodies that bind particularly to the mast cells. Remember, in the inflammatory response, we have mast cells releasing things like histamine, and those histamine molecules and those mast cells are going to have an IgE antibody bound with them. These are going to be particularly effective in allergic reactions as well as in defense of things like fungal and parasitic worm infections. Here's the graph I was talking about. When you have your first antibody response, a first exposure to a particular antigen, you have something called a latent period. And after that latent period, you're going to begin to produce IgM antibodies. And you don't get a whole lot of those very rapidly. This latent period can last probably between 14 and 21 days. So you are certainly going to get sick in this latent period. Once you have the IgM antibodies produced, then you're going to start producing these IgG antibodies. You have a greater number of these, but you'll notice that the IgM begins to wane as the IgM begins to increase. You have a relatively high total antibody titer, but again, you are going to get sick because this antibody in circulation is going to be very slow in coming. You have some interval of time, you've gotten better, but then you've gotten exposed again, and you have a second exposure to a particular antigen. Our immune response is called an anamnestic response. If you think about what does amnesia mean, amnesia means to forget. So anamnesia means to not forget. So we have a memory response. So once we have this second exposure, we have an almost immediate production of IgM and a very high production of IgG. You'll see that our IgM begins to wane off a bit, but our IgG climbs, and it does come down a bit, but it then begins to level off. And so we have this very high total antibody titer at the end of our second, third, fourth, 100th exposure, and we begin to build what we call titer. And our immune system then recognizes this response, and we get this huge IgG antibody response. And these antibodies are going to last for a much longer time than they did in our primary exposure. IgM is still going to be produced first, but the IgG is going to continue, and it's going to last a much longer time. So we said that you would have that latent period. You have no antibody synthesis here, but once you have that antibody synthesis, you're going to have that titer begin. IgM is going to be first, IgG is going to be second, and you may also have some IgA as well as a little bit of IgM coming to the response again.